Hello guys, welcome back. All right, so um, this is a new lecture, and we are going to talk about the blood. So this is kind of a transition from our earlier lecture in the cardiovascular system. We talk about this organ system, um, very like um, visible, right? It's like heart, pump, blood flow, and this system uh, the function is to transport the blood. So now we are going to see what's this thing, the blood. You know blood, this is not new to you. What we are going to learn is basically the function of the blood. So in order to know the function of blood, we need to know its components. And so we can kind of tease out what's in there. And so we can know that what is the function. All right, so the first is that, okay, let me see that. So first is that when you get the blood, we can, the whole blood is red, a lot of fluid, right? It's fluid and a lot of stuff in there. So, but it's a combination of so many things. So what we usually do the first is that in order to know the components, we can try to separate. You can separate things by different way. A very common way is to do like a centrifuge. So basically that here, centrifuge. So basically that you can we will do this in the lab. So we have something, this is called a capillary tube. Um, we will know more in the lab, but basically this is a glass tube, very small, and you will get if we get a little bit drop of the blood, fill it in that, and then we can put it in the centrifuge in a way that okay, we put it there, and if we just close it up and let it spin, if we let it spin for a while, we will be able to separate components of the blood into lightweight components such as water. Those are pretty light, or heavy weight stuff. So that will be the red blood cell, white blood cells, those large. Those are heavy stuff. So we can separate it. So this the first step. We are going to know the components of the blood. So by doing so, we can separate it. So we can see that there are two examples, A and B. Basically, one is from the male. It doesn't really show here, but one's from the male. The other one's from the female. Male has more red blood cell in the blood. Female has a little bit less red blood cells in the blood. All right, so this is the situation. So that's what we got first. By doing the centrifuge to spin the blood sample, we can separate the watery portion and also material portion. We call this watery portion a plasma. So that is a plasma. We call this material portion, we call it formed element. It doesn't show here, but this one is called formed element. What we also see very commonly see is that we can get a portion that is the dramatic difference that you can see here is that you have a segment that is red and a segment that is transparent. So this red segment, we call it, this is composed of compact red blood cells. So we call this is the percentage of this red blood cell among the entire sample. We call it the hematocrit. So that's that. So the first terminology we learned today is hematocrit. What is hematocrit? The percentage by volume of the red blood cells in the blood. So that's that. All right. So there are, even though plasma is transparent, it's not pure water. Okay, so your plasma is not pure water. So here is the components, break it down. You have the plasma right here. And in the plasma, this fluid, we call it plasma. We have water, large portion of those is water. We have protein, and we have some other water-soluble stuff in there. So in addition to the protein, those are also water-soluble. And so in the protein, you need to know that there are leading like most abundant plasma protein. Those are in the blood, but they are not in the blood cells. They are in the plasma, in the fluid. So those proteins, the number one, the most common one is called albumin. All right, so we're gonna have quiz on that, which is the following is the most abundant plasma protein. All right, so albumin. The second one is globulin. So the globulin has many things. It's a collection of so many things. A lot of those basically is just carrier proteins. Yeah, so and a lot of those also be antibody. It's called immunoglobulin. So they are a collection of so many things. The third one is important though. The third one here is called fibrinogen. So fibrinogen is associated with our bleeding. So this one is very important to stop bleeding. So you can see that it's something gene, right? Like earlier we talked about angiotensinogen. So those are the inactive form of that protein. So they are not do the work, but they are ready to be converted to become an active one because we don't want to get blood clot very easily, right? So these are the protein that floating around, but they are very non-active, so they don't form the blood clot. But once there is an injury, we can convert this fibrinogen to become fibrin, then they will form the blood clot in order to stop bleeding. So that's the plasma protein. There are a lot of proteins in the blood. We can use it to do so many different diagnoses. These are just the most abundant top three, okay? And this is the one that you got to know. People talk about the albumin, and we also use, so that's that, right? And in this portion is called the formed elements. So formed elements are basically packed of blood cells. So from here, you can see that we have three types of the blood cells. The majority of the blood cells are red blood cells. So that's the most abundant blood cell. But we have red blood cells, white blood cells, platelet. So there are three type blood cells, and all of them combined provide these formed elements. So that's that. So you can see that we have platelet, we have white blood cells. White blood cell is a collection of white blood cells. White blood cell is also called leukocyte. Later we will review that. It includes 5 major type of white blood cell. You need to know this though, but later we will also emphasize on that. Neutrophil, that's the primary white blood cells. Lymphocytes, monocytes, eosinophils, basophils. So those are the white blood cells. Red blood cell is very homogeneous, very consistent. We only have one type of red blood cell. That's just red blood cell. It's also called erythrocyte. We will review that later. So that's the components of the blood. All right. First, you need to know there's plasma. When people talk about plasma, basically is the components that doesn't contain the blood cells, which the following is not part of the plasma. Don't choose. It's not, right? So those will be, blood cells are not part of the plasma, which the following is part of the plasma. So that will be these protein, albumin, globulin, and the fibrinogen, right? Which of following is the white blood cell? Neutrophil, lymphocytes, etc. Which of following is the red blood cell? Erythrocytes. Yeah. All right. So this one just basically just summarize what we talk about from the picture. Just present them as text, right? So we have the blood has if we do the centrifuge, we can separate them as plasma, the fluid. This fluid is also called extracellular because they are outside of the cell, right? So it doesn't have the cells, outside of cells. Our body has a lot of water. 70% of our body's weight or volume is water. And those 70%, part of them is in the extracellular, part of them is intracellular. In the cell also has water. Yeah. Plasma has a protein. Later here we talk about the plasma protein, albumin, globulin, fibrinogen. All right. Plasma also has ions and other solutes. Formed elements are cells. We have red blood cell, erythrocytes, white blood cells, leukocytes, and platelets, thrombocytes. So that's that. And the proteins are albumin. You don't need to remember the number, but you need to know which one is the most abundant. So it's always location and function, right? So where is plasma? Where is albumin? In the plasma. What does it do? It has many functions, but the major function here we provide here is to maintain the osmotic pressure. Earlier we talked about that isotonic, right? Your blood, your body fluid is isotonic. Then what contributes to that isotonic? The major components to maintain it is the albumin. So, but it's just the one of the function. It has multiple functions. Globulin is also a collective group of many different proteins. A major one we talk about is antibodies. It is also called immunoglobulin. It also serves some other globulin, not the immunoglobulin. Some other globulins also serve the transport protein. Why do we need transport protein? Because a lot of these components in the blood, we need to transport it. We do transport oxygen, but oxygen is not one of the plasma protein though. Oxygen is transported by hemoglobin. It's globulin, but it's in the cell. It's in the cell, not in the plasma. So that's different. But same thing, you know, we also transport several other things. We transport iron. We transport a lot of proteins. One thing is called the LDL, low density lipoprotein, right? So those are transporting lipid. So we have different kinds of like carrier protein to transport materials throughout our body. Fibrinogen, we talk about that, stop bleeding. So that's their function, location and function. All right, so all these cells, you know, we have so many cells in the blood. Almost 50% of our blood volumes are cells, right? Almost, not yet, not exact, but almost 50% of the blood volumes are cells. Then where are these cells come from? These cells, all of them are originated from the stem cells in the bone marrow. So this one I want to show you. So, okay, blood cells from the bone marrow. All right, so this one just, we will talk about the bone marrow right here. Next one, I think. All right, so blood cells, function, all right, location and function, all right? So blood cells, what location is in the blood. And later we will know that they are originated from the bone marrow. Just hold that thought a little bit, because we, like 2 slides later, we will talk about that. But what's their function? The major function for the red blood cell is to do oxygen transport. The major function of the white blood cell, you have 5 major members: neutrophil, eosinophil, basophils, monocytes, lymphocytes. Their major function is body defense. The terminology here is called immune system. So immune system is the next lecture, we will focus on this one in our next lecture, talking about the immune system. And white blood cell is a major cellular components in our immune system. And platelet, what's their function? Is to stop bleeding. It's another terminology here. It's called hemostasis. So yeah, so that is the hemostasis, is stop bleeding. I just want to, before really talk about that, I just want to remind you that it's different from homeostasis, okay? Because I figured that in my like earlier lecture, people asked me about that. What is It sounds so similar, but it's not the same, okay? Hemostasis is the one that we talk about right now, is the process to stop bleeding, cessation of the bleeding from the blood vessel. So it's not, hemo means blood. It's not home, right? Homeostasis is home, right? Your house, your home. Homeostasis is the one that we talked the very first lecture. In the very first lecture, that's homeostatic loop. So maintain a steady internal or environment, you know. Yeah, so that's that. So that's the difference. So don't get confused. Hemo is blood. Homeo is home. So hemostasis is the one that is all stable. Hemostasis is one to stop bleeding. All right. So all these cells. So as I talked about earlier that all these cells come from the bone marrow. So they all come from the stem cells. This stem cell is called hematopoietic stem cell. And then this hematopoietic stem cell, they are stem cells. So they are in a particular niche. That environment help them to reproduce themselves. Yeah, so that's that. So they are in the bone marrow. And then they will get, once they are proliferated, meaning that they, duplicate themselves to multiple, they will gradually differentiate process to become different. I think we kind of talk about it, right? Proliferation, differentiation. So differentiate is to build different characters for that particular fate of a cell. Yeah, so they were differentiated while maintain some proliferation capability. Yeah, so they will first differentiate into two type of stem cells. So these two types of stem cells, they are still stem cells. They can reproduce themselves, but they start to show some characters that different from the other. So they differentiate into the myeloid stem cells or lymphoid stem cells. And then lymphoid stem cell will gradually differentiate it into lymphocytes. Yeah, and lymphocytes have three types of lymphocytes: T lymphocyte, B lymphocytes, and the natural killer cells. We will talk about that a little bit here, maybe, but we will talk more in our immune system. So this function is to deal with the immune system. Myeloid stem cell is the one that will further differentiate it into all different other cells, differentiate into the mast cells. So this is not a blood cell. Mast cells, once it's differentiated into these mast cells, will moving into the tissue. So it's part of our immune system, kind of a surveillance, like a agent to in the front line to detect any intruders. So that's the also differentiate from the myeloid stem cell. It will differentiate to other leukocytes, basophil, eosinophil, neutrophil, and the monocytes. And so those include the lymphocytes. These are white blood cells. It will also differentiate into the megakaryocytes, and this one will provide the platelets. And the one that we are focusing right now is the one that's red blood cell or erythrocytes. This one is also differentiate from the myeloid stem cell. So just get you have an understanding of that these blood cells, including the one that we will focus on, that's a red blood cell, that's an abundant one. The most abundant cells function is to transport oxygen, but we also use the bone marrow's hematopoietic stem cells to derived to so many different kind of immune cells, white blood cells, lymphocytes, etc. So that's that. Everything comes from here. Yeah. Right. So the quiz question can be that which of the following cells are derived from the myeloid stem cell? Basically, everyone is except the lymphocytes. And which one is derived from the lymphoid stem cells? That's the lymphocytes. A quiz question would be that where is the hematopoietic stem cell? Hematopoietic here. HSC. Where is it? It's in bone marrow. So that's that could be the question, right? Yeah. So that's that. So cell production is called hematopoiesis. So this one just hemato means like blood. Hemo is also means blood, like hemostasis, right here. Hemo, right? Hemato, hemo, all right. So cell production, blood cell production is hematopoiesis. So this basically describe, especially here, hematopoiesis, blood cell production. And the origin, basically the mother of all these cells is called hematopoietic stem cell right here. Hematopoiesis is process. Hematopoietic stem cells, HSC, is the stem cells for so many different other cells. Yeah. Hematopoietic stem cells in the bone marrow, they can differentiate into two category, two type of the stem cell, myeloid and the lymphoid. Yeah, and then we know that lymphoids produce lymphocytes, and every other cells come from the myeloid stem cells, right? So that's that. So I think that's everything we need to know about the blood, all right? So in this lecture, so we can talk about, we can talk about all of them. You know, they are all very important. They are the major cells in the blood. Red blood cells, white blood cell, platelets, and that's why we are going to only talk about these two in this lecture, and we will focus on these immune cells in our next lecture. Yeah, so we will focus on this one. So first, we will talk about the red blood cells. For the red blood cells, I want to talk about it in 4 different portions, and they are all very. I just want to organize in a way that you know that this is what we talk about, that's what we talk about, you know. So, and they are not very deep. You know, I try to just cover the essential portion. You just need to know this, and you're good. Yeah. So, right. So the first one is to talk about the origin. Kind of know already, right? We know it's from the blood, from the bone marrow. Myeloid stem cell produce erythrocytes. So we know that already. We just provide you one extra fact, one extra component to that process. And then we talk about function. You guys know it already. The major function is oxygen transport. We just want to add one more components, one more details to it. All right, so it's fair, right? It's not super complicated. We just add a little bit more. All right. So but you guys already know, you know the function, you know the foundation. We just need a little bit more. Now another thing is that degradation. So we usually don't talk about cell dies. You know, it's a funeral in a way. That yeah, we talk about its function. When we talk about the cells, we talk about organs. We always talk about its function. We talk about location and function. We don't usually emphasize its dying. We do for the red blood cells is because they are abundant and they have a short lifetime. Okay, so it's something that we need to deal with. Our body needs to deal with. We just need to have a mechanism to deal with these recycling in a way that cell death. We have abundant cell deaths every day. Yeah, how do we deal with them? Yeah, we don't really have that problem with the nervous system because nerve cell never die. You have the same nerve since your first day in your life. Muscle, basically the same thing. Heart cells, once it is differentiated into heart, it's the same heart. So take good care of them because you are not going to regenerate them. Skin cell do die, but skin cell is easy. They just shed away. You know, it's outside, so you don't need to worry about it. Yeah. But the red blood cell is abundant. I don't know. I don't know whether that is the most abundant cells. We do know that we have a lot of blood. We have a lot of blood cells. We have, and the red blood cells most abundant. So we talk about it. Every RBC only have about 4 months of life. So it's limited life, and they will die. And how do we deal with them? So we talk about that. Finally, we will talk about the blood typing. A type, B type, AB type, O type. So that's that. All right. So let's start by talking about these one. That's easy. All right, let's take a look. So origin, bone marrow. You know that what stem cells are they produced from? Myeloid stem cell, right? So we know that, right? So it's here, right? Myeloid stem cell, right? So we know that, all right? So just something we know about, just give a closer view about the red blood cells since we are going to talk about it in this lecture. So red blood cell is also called erythrocyte. I may have that quick question, but I feel like it's so naive, so I hope it's not like insulting your your understanding. Which of cell is erythrocytes? White blood cell, red blood cell, something like that, right? So it's red blood cells. Yeah, erythrocytes. All right. Yeah. So this is most abundant blood cells, and the shape is a disc shape. All right. You don't need to know the number. You just need to know that that shape represents one important factor, which is that these cells do not have nucleus. Okay, so it's if you look at the microscope, we are going to use microscope in lab, and you will see that all these a little bit hollow in the center is red blood cell, and you don't see the nucleus. If you look at other cells, later we probably will see other cells. You will see they have a nucleus, but not red blood cells. You don't see nucleus. Yeah. All right. So it's a nucleate. So it doesn't have a nucleus. So once it differentiates into the matured one, the nucleus is degraded, is removed. So that's that. So these function, we talk about the function of red blood cell. Its function is to transport oxygen. And we know that it's produced from the bone marrow. And the thing is that how do you know how many red blood cells you need to transport oxygen, right? It's important that this oxygen is not just needed by one cell, it's needed by whole body. So your body need to tell bone marrow that, okay, please produce more or please produce less of the red blood cells in order to accommodate the whole body in that environment. And so here I want to tell you is that the messenger to tell the bone marrow to produce red blood cell is erythropoietin right here. Erythropoietin. It comes from the terminology here, erythropoiesis, right? Poiesis is hematopoiesis. I'm like teaching English right now. Hematopoiesis, what is that? Hematopoiesis. Poiesis is creating and making new cells. So blood hematopoiesis, hematopoietic stem cell. So this is the process to produce the blood cells. But if we are talking about the red blood cell, it's erythrocytes. So it becomes the erythropoiesis. Yeah. And the factor to stimulate the erythropoiesis is called erythropoietin. So that's easy, right? Which of following stimulate the bone marrow to produce red blood cells? Just by a name, you know which one is which, right? So it got to be red blood cell. It got to be production. So it's erythropoietin. So that's that. We have in our body, we will tell bone marrow, hey, we need more oxygen, right? This is the Mars. We don't have oxygen, something like that, right? We don't go to Mars, but we do go to like higher altitude or higher, higher mountain, right? High mountain. Okay, how about that? High mountain. We do go there. Some people live there. People live there, or yeah. So there will be situation that you need to accommodate into that low oxygen environment, and in order to transport your body with more oxygen, so you will tell your bone marrow to produce it. Erythropoietin. So that's a new terminology, even though this terminology is so direct to tell you what it is, right? Just like angiotensin, angiotensin is blood vessel constriction. Erythropoietin is erythropoiesis in, right? So it's an ingredient, right? So it's erythropoietin. So what's its function? Location and function. What is its function? The function is to tell bone marrow to produce red blood cells to conduct this erythropoiesis. Now, location. Location has two, right? Where is it from and where does it act on? So where is X on? It acts on the bone marrow. Then where is from? It's from kidney. Kidney is such a interesting organ. It's like it wants to get involved in so many things, right? Earlier we talked about the renin-angiotensin pathway, RAS, right? Renin comes from the renal, is from the kidney. Now erythropoietin also comes from the kidney. Why does kidney get to involved in oxygen sensing? It's because that kidney is an organ that doesn't really use that much of the blood, but there's a lot of blood going through. It's a high traffic area, a lot of blood going through. Kidney's function is to clean the blood, so a lot of blood going through. And so this is a perfect sign that if your blood oxygen level is low, it knows, it senses it. If your blood pressure is low, it knows, it senses it. And so if your blood volume is low, it knows, it senses it. And that's why it's involved in the cardiovascular system and involved in the blood production, red blood cells production. Yeah, so that's that. Kidney produce it. So if the environment is, tend to have low oxygen, over time it will stimulate the kidney to release erythropoietin, and then erythropoietin will act on the bone marrow to produce red blood cells. And so red blood cells will transport more oxygen, and so it will bring up the oxygen level in the body. Yeah, so that's that. That's the only detail you need to know about the erythropoiesis, the production or the origin of the red blood cells. Easy peasy, right? Now, the next thing is the next topic about the red blood cell is the function. You already know its function is to do oxygen transport. So we just want to add one more details to it. Yeah, so it's baby step. We can do it. All right. So what is it? what do we need to know about oxygen transport? One thing is that oxygen doesn't get dissolved in the fluid. We all know that gas, like if you just blow air into the water, they just evaporate, right? They don't get dissolved in water. It's different from if you put some salt into water, they got dissolved, right? If you put some sugar in the water, they got dissolved. Gas is different. If you blow a lot of gas into the water, they just get just leave, right? So we need something to capture the oxygen in order to transport them so they don't just get leave. And that's the thing. Red blood cell is just a cell, and its function is to carry oxygen. But what is that thing to really capture the oxygen in the red blood cell? The thing is the one that we are going to talk about. This thing is called hemoglobin, all right? Hemoglobin is the protein inside of red blood cell. Location and function. Where is hemoglobin? It's not in the plasma, okay? Red blood has hemoglobin, but hemoglobin is not in the fluid. It's not in the extracellular fluid. It's inside of the cell. So it's right here, cell, inside of cell. So when cell differentiated into different cells, we talk about that, define the calling. How do we know it served its calling? We see what it produced. And red blood cell, when it serve its calling, it produce what? It produce hemoglobin. This protein from gene expression, DNA to RNA, RNA to protein. We talked about that in our earlier lecture. And that gene get exon, and these cells gradually become red blood cells. That's its job, produce a lot. The whole cell is packed of this hemoglobin. One thing I want to talk about this hemoglobin is go into more detail. You see this several how do we call it? Turn? This kind of thing. These are represent the protein. As I told you earlier, that protein, once it's produced, is a polymer of amino acids. So we have the primary structure. It's not functional. We need to fold it. Okay, so this is folding. We need to fold it. And we need to fold it in a way that it serves its function. Hemoglobin has 4 units, and each one is very similar. So we have 4 identical, not exactly identical, but very identical units. Each one is a pocket. So it folds this amino acid polymer, folded to become a pocket. What do we do with this pocket? In this pocket, it pocket one component. This is not, and the thing that get packed in that chain, we call it 4 chains. A thing got packed in that chain is called heme. So these is a heme. These is a heme, and we expand it. You can see that these is a heme. Heme is a chemical structure, and it got packed into each chain of a globulin. So what is a hemoglobin? Hemoglobin is a protein synthesized by these red blood cells, and it has 4 chains. So basically, you have a gene produce amino acid polypeptide, and a polymer, amino acid polymer. We call it peptide because it's not folded. Then we fold it to become a little pocket, and then 4 of them combine together to become the tertiary structure, right? You have the primary structure, secondary structure, tertiary structure. Quaternary structure is one that's what we see here. So that become the combination of 4 different chains. Yes, 4 different chains, and then each chain contains heme. And this is how we capture the oxygen, because in the heme we have an iron. Oxygen can attach to iron, and so each hemoglobin can carry 4 oxygen maximal. If yeah, so that's that. So that's hemoglobin. That's what you need to know. So each hemoglobin can carry 4 oxygen maximal, and when, but how do we, and if it carry all 4 oxygen, we call it fully saturated. If your blood, every hemoglobin are all packed with 4 oxygen, then your saturation percentage will be 100%. But sometimes we only have a situation like it doesn't really carry to its potential, right? Maximal potential. We then have a less than 100% saturation percentage, and we can measure it. We can measure this saturation percentage by this. This is called pulsoximetry, meter, pulse oximeter, and we can measure it. So when we measure it, basically we can measure pulse. Basically, it's pulse rate. We can also measure the oximeter, the oxygen saturation percentage ratio. And so you can see that this is 99 is the saturation percentage, 79 in this case is the heartbeat. Yeah, so you always get 2 numbers from, we put this in the finger, and yeah, so we can measure that. All right. Then the next thing I want to tell you is that what determines its saturation percentage. And right here. So this is one we are going to talk about. This is a curve, we call it oxygen dissociation curve. So in this curve, we have X, any curve, we have X and Y. So let's take a look. So what do we have here? So the X-axis is oxygen partial pressure. So we talk about the, so this one, we want to see that basically this curve has X and Y, right? So X-axis is oxygen partial pressure. So this one is referred to that the environment, how much oxygen is there, and how much is that using the unit of the pressure. So how much pressure provided to the from the environment is from oxygen. So let me give you an example. For example, let me see if I can get a pen and pencil. Sorry if I can. All right, so let's do this one here. So let's do this one here. All right, so here we have, let's see. All right, so we do have that. Good. All right. So let's say that the air, right? This is the atmosphere, right? So what is the pressure of the atmosphere? The pressure you probably remember, which is 760 mmHg. So that's the atmosphere pressure. Yeah, so let me show you this one here. So we have, let me show you this one. So here is the atmosphere. So atmospheric pressure. So this is the environment on the Earth. So you can see that the pressure just right here. So it's a 760 mmHg. So this is the basically the pressure from the air, right? So that's that. All right, let me go back to the see that so I can minimize it and also get some annotation. Good. So that's the atmosphere. Nice, nice. All right, I have it. Yes. All right, that's all good. All right, so it's 760. Let me see that. So it's the atmosphere is 760 mmHg. So that's in the air. Now, in this air, we know that it's not all oxygen. So oxygen is just partial of that air. The oxygen is about 20% of the all air. So we times 20%, right? So times 20% divided by 100. And this one will give you a number which is about 150 mmHg. So this will be the pressure from the oxygen. So the total pressure from the air is 760, but the oxygen partial pressure is 150 mmHg. So just give you an example that what does it mean by oxygen partial pressure? So we can see that this is the situation that is in our environment on the Earth, right? So with this one, you will have partial pressure of 150 mmHg. So you can kind of imagine that even though you don't feel the pressure, you don't feel the air pressure, but in fact, that is constant pushing the air on you. So you carry those pressure on you. You just a climate to it. You just feel you just live here for so long that you don't feel that pressure. Okay, so that's that. Now with that pressure, 150 mmHg, that pressure pushing on you, pushing through your air tract, pushing through your trachea, bronchial into your aorta. Your aorta cells, the red blood cell in the aorta, experience these large pressure, these large pressure. These large pressure, 150 energy. Then you can ask that with that pushing, how effect do hemoglobin receive those oxygen? And so you can see that with that pressure, 150 right here. So here is 100 mmHg. So the 150 will be somewhere that we can extend it here. So it's somewhere like here, right? So 150 will be something like here. 150 mmHg. So with this one, you can see that this curve we extend here, it would be somewhere like this. This is already reached the maximum, 100%. So with that pressure, with the normal air that you breathe in, this huge pushing will push all the oxygen get bound to hemoglobin. So with this one, we will have 100% saturation percentage. So with this one, you push the hemoglobin, you push the oxygen, push it because pressure is so high, push it. And we can remember that even if you don't get the 150 mmHg, if the oxygen is lower, you still be able to push it. So in our body, this hemoglobin is the best, is a design that we can live on Earth perfectly. In the air, you don't need to make any much effort because the air, the oxygen level, the oxygen partial pressure, the property of this hemoglobin will allow this oxygen get bound on it. So very easy that if you don't have any disease, this oxygen bind on hemoglobin, you can get 100% saturation percentage. So you do your oximetry, you will get nearly 100%, 99, something like that here, 99. It's because that when the oxygen received in the alveoli is circulating from the left atrium, left ventricle circulating. When they circulate into your finger, you already use some. So it's not 100 anymore, but it's still near. You have good amount of oxygen in your blood. So that's that. All right, so let me clear it. So that's the foundation of it. Let me see if I can. Okay. All right, so that's that. So that is it. All right, so we have that. All right. All right. So Okay, all right, so that's that. So this is this curve. So so this is in the lung. Basically it's in the lung, you are in the air, then your hemoglobin saturation will be 100%. Now, when this blood going into your tissue, they are going from the high oxygen area in the lung, moving from the aorta and moving to the tissue. When they go into the tissue, when they go into the tissue, they are going into the place that oxygen level is low. Why the oxygen level is low? Because in that tissue, in your muscle, in your organ, those, if oxygen to cells, so your cell, muscle cells, brain cells, all your body cells, they need oxygen. They need oxygen to produce ATP. So when oxygen, if is oxygen to those cells is very much like money. You like money, right? So it's like in the room, right? If you see any money on the floor, what you will do, you pick it up, right? You take it. Cells is the same thing. When they see oxygen, they will use it because they need oxygen to produce their ATP. And so in the cells, it's a very different environment. It's not like environment. It's not like air. In the cell, in the tissues, in our bodies, tissues, the oxygen level is very low. So with that low level, so oxygen partial pressure will drop to about 40 mmHg. And with that, you don't maintain that huge push of the oxygen to bind on hemoglobin. That push become much less. And with that environment, your hemoglobin cannot hold on that oxygen anymore because outside doesn't really have that oxygen pressure. So hemoglobin cannot hold on it anymore. And so hemoglobin lose that holding. And basically another way to say is that oxygen doesn't have that strong affinity to hemoglobin anymore. So oxygen will evaporate out, will leave. And by that leaving, they are free and they will be captured by the cells. Cell will use them to produce the ATP. So you can see that in that environment, the hemoglobin concentration and saturation percentage will drop to about like 75-ish. Basically, you can see that in the beginning, each hemoglobin can find 8 oxygen, not 8, 4 oxygen, but each oxygen is O2. So you have 8 total oxygen atom. But each one can carry 4 oxygen, right? But now you lose one, you have the other 3. And so your percentage is not 100% anymore. You drop like a quarter of it. So you become like 75-ish. Yeah, so that's that. And when you exercise, say if you're exercising, then your muscle use more oxygen. And so the environment become very hypoxic. And so not very, but kind of hypoxic. So when you exercise, you basically create your body's environment to be hypoxic. You give your body some hypoxic stress. And with that one, the oxygen level is very low. And so the environment doesn't really have a lot of oxygen. So more oxygen got evaporate out from the hemoglobin. And so your saturation percentage will become even lower. It will become less than like 30%, something like that. So this is a curve. This curve give you the idea. Basically, this curve give you the idea that what is a primary factor to control hemoglobin saturation percentage. The primary factor is this one, oxygen partial pressure. So what regulate it? What control it? How do you make it bound? How do you make them bound on hemoglobin? How do you make them unbound from hemoglobin? The way we regulate it is the environment. The more you push it, they got bound. The less you push it, they got unbound. So that saturation percentage. The primary factor to regulate it, to affect it, is oxygen partial pressure. So that's that. All right. Yeah. There are many other factors can affect it, but those are not as strong as the oxygen partial pressure. Oxygen partial pressure is very strong. It's the most important one. So that's that. Now, knowing all this, all right, so here we are talking about the function of the red blood cell, which is oxygen transport. And so we talk about hemoglobin. Hemoglobin is that molecule, that proteins to bond to do that work, to bond the oxygen. Now, the other thing that in addition to know the saturation percentage, another thing is that, well, how much hemoglobin do you have? That's the question. How much red blood cell do you have is a different story though. How much red blood cell do you have is hematocrit, right? So this is the one that the volume right here, the volume of red blood cell is hematocrit. But each cells can have a good amount of hemoglobin, right? This is a bag. Some of them has a lot, some of them have less. So do you have a good amount? Even though your red blood cell is very, you have good amount of red blood cell, but do you have good amount of hemoglobin in it? If you don't, you may have anemia, right? Anemia. Yeah, the hypoxia in a way, right? Anemia. Your red blood cell doesn't carry enough oxygen for you. Yeah. So do you have enough hemoglobin? And there are several ways to measure it. In the lab, we will try one method to measure. So we can measure red blood cell by doing this hematocrit. This one doesn't give you the idea about hemoglobin. As for the hemoglobin, we use another reagent. So this is the one that we are going to talk about right here. This method is called Telquest table, right here, Telquest paper. So we're gonna have the paper, we're gonna drop blood on it, and then we're gonna compare the color of the blood with this table. And so the normal range of the hemoglobin is about 15-ish, so it's about like 15 grams per deciliter. Yeah, here, 15 grams per 100 milliliter or deciliter. Yeah, so that's the normal range. It's different from the hematocrit. This is the hemoglobin weight. So you have that good amount of hemoglobin. And yeah, so that's that. You just need to know what method we use. Nowadays, this is for lab, we do it manually, but nowadays you basically just would do a complete blood count. Some of you probably do know this one in the hospital, CBC, complete blood count. It will give you everything. But in the lab, if you don't have that device, nowadays basically all hospital has that device. But if we don't, we can examine it by this Telquest test. So that's that. All right. Let's take a break, and we will move on to the other topics of this red blood cell. All right.